And we are going to move on with Dr. Alpha Lee from the UK.
Thank you very much. My name is Alpha Lee. I'm a group leader in the University of Cambridge in the UK. Our group focuses on using machine learning and artificial intelligence to find new drugs. The challenge with modern drug discovery is that it is too costly and too slow. The time it takes to find a new drug, as well as the cost of drug discovery, has exponentially increased over time. It now takes billions to find a new drug, and the process can take as long as 10 years with an overall 4% success rate. The reason why it's getting slow and costly is that molecules in nature that can be readily repurposed as a drug has mostly already been found. In order to treat diseases that cannot be treated in the status quo, we need a new way to find and use more complex organic molecules. And trying to make everything under the sun and testing every single molecule that we can make is simply not a viable strategy. We need something quicker and cheaper. And the idea is simple. Suppose we test a small number of molecules against a particular biological target. And out of these small number of molecules we tested, some of it will work, some of it won't. We then develop an algorithm, a computational tool that can tell us why the molecules that do work actually work, and then we can build a computational method that can replace experiments and screen a lot more molecules in a lot shorter time. Let me give you an example of what I mean. The 4 molecules I've shown in this slide on the left are molecules that can activate a receptor known as ADRB1. It is important for modulating heart rate. If you look at the 4 molecules on the left, you might realize that they all share the group that I circled in orange. This might then therefore be a pattern that is recurrent in those 4 molecules that causes it to work. But if you look at the figures on the left again, you might realize that they also share the motif that I've circled in green. Now, circling things is okay if you have 4 molecules. But what if you have 400 molecules or 4,000 molecules? How can you identify motifs, chemical groups that are actually therapeutically relevant versus other motifs that arises simply by chance? And that's exactly what we developed, a computational tool inspired by machine learning and pattern recognition, which allows us to identify chemical structures that are therapeutically relevant, and then building a model that can replace experiments and can identify which unknown molecules would also be biologically relevant and useful in the body. We've actually tried deploying this algorithm prospectively in a problem that is interesting in medicine. We focus on the so-called CHRM1 receptor, which is a receptor responsible for symptoms of Alzheimer's disease and schizophrenia. We took 6 million commercially purchasable molecules. We used our software to narrow it down to 100 candidates, which we then experimentally tested. And out of 100, we found 4 novel and active compounds. And the hit rate, the success rate of this algorithm is twice more than the state-of-the-art in the industry. And of course, testing millions of molecules is not possible, but testing hundreds of molecules is definitely possible. We can also learn the language of chemistry using machine learning. And the idea is that we can realize a new way or new ways of making complicated organic molecules by treating chemistry as a sentence. It's not showing up in the slide, but please come to my poster. And we treat reactant and reagent as a sentence, like a language, like English, and the product as another language, like Swedish. And we use the technology of Google Translate to predict what is the product given reactant and reagents. And surprisingly, the algorithm can learn from scratch without any human supervision from openly available chemical data, achieving an accuracy of 90%, and even a well-trained human chemist can only achieve an accuracy of 75 to 80%. So an algorithm can autonomously learn chemistry from scratch and achieving accuracy above what you would expect from a human chemist. In summary, machine learning and pattern recognition provides us with a tool to accelerate drug discovery by allowing us to identify useful and interesting molecules out of a large haystack of possibilities, as well as finding new ways of making complicated molecules that's never been made before. Thank you very much.
Thank you very much. Is there any question? Yeah, up there.
Hi. How do you select the molecules that you want to put into this algorithm to develop new drugs?
This is a great question. So we take molecules that already have been screened for biological activity. So we leverage on historical high-throughput experiments, which generated some data on, let's say, 1,000 molecules that people have screened. And then we train the algorithm so that algorithm can predict which other molecules will also work. And in terms of how to select these initial molecules, usually we select it based on being different to each other, so that you sample a lot of different chemical motifs rather than focusing on narrow region of chemical space, as well as we want to focus on molecules that are drug-like rather than known to be toxic or known to be bad for the body. So we want to focus on first drug-like molecules that are chemically diverse, so that we sample a wide region of chemical space.
How, if you just sample molecules that already have a certain effect, how will the new molecules that you discover be any different and have a different effect than the previous ones?
That's a great question again. So this algorithm predicts whether the molecule works against one particular protein receptor. But ideally, for a drug, you want a molecule to work against one protein receptor but not others. And that's where the novelty comes in. So the algorithm, based on the data, the data that you have only tells you that this molecule works for A, another molecule works for B, another molecule works for C, but not really solving the multi-objective optimization problem of finding a molecule that works for A but not B and C. And that's where the algorithm will be able to do it. It puts together bits of the molecule that works for A, but not the bits of the molecule that works for B, but perhaps the bits of the molecule that works for C, together to form a monstrous molecule, which no one knows how to make now. And then the other algorithm that talks about the translation algorithm tells you how can you make it using commercially available building blocks.
Okay, thank you.
Thanks.
Thank you. Okay, thank you very much.
Thanks.
If there's no other question, okay, thank you very much.
Thanks.
And